This video shows you how to analyze the group read live table. Can toSeq analyze group read live table data? If insects are reared in groups, you can use toSeq to analyze your data and calculate the population parameters. Your data must be collected using the same time unit throughout the whole period of live table study. Before you carry out group read live table study, you must detect the proper density for group reading using a few preliminary studies. Record the group read live table data as this table. When you collect the live table data using group reading, you keep insects as a group. In general, you keep individuals of different stages in separate containers. You can record the number of surviving insects in each stage and daily total fecundity of all females. At H0, you begin with 100 eggs. On day 9, there are 72 larvae and 26 pupae. Note well, there are 51 females observed on age 14 days. They have laid 129 eggs. You should know that you will see these 129 eggs on age 15 days. Keep this in mind when you prepare your data file for analysis. In the group read live table data table, the daily stage structure forms the matrix and the daily total eggs laid by female adults forms the matrix F total. The daily stage structure is actually the matrix and the daily total fecundity is matrix F total. On day 9, there are 72 larvae and 26 pupae. The 129 eggs observed on age 15 days is the age-specific fecundity of females of age 14 days. They have laid 129 eggs. You can calculate matrix S from matrix N by dividing all elements of matrix N with N0, 1. For example, in matrix N, there are 72 larvae and 26 pupae on day 9. They divided by 100, the survival rates are 0.72 and 0.26 for larval and pupal stage respectively. You can calculate fecundity matrix F from matrices N and F total. Each F total is divided by the respective NXJ to get the FXJ. For example, at age 14 days, there are 51 females. They laid in total 129 eggs. The mean fecundity is 2.5294 eggs per female. Population parameters can be calculated from matrices S and F. For a better understanding of live table theory, please read following references. The first and the second papers have been cited more than 1,000 times. The third and fourth papers are comprehensive and important. Based on the above discussion, you know that you can calculate population parameters based on group read live table data. Therefore, if it is impossible to rear insects individually, you can rear them in groups. You can still obtain matrices N and F total. You can then calculate S and F. Then you can still get the R, R0 and T as if you had kept them individually. This slide shows you the data format of group read live table for toSeq program. The first 3 lines are descriptive information. The third line is the treatment code. Please read this slide carefully. If you work with parthenogenetic population, there is no male. But you have to prepare the data file with the male line, male, 0, 0. If you collect the group read live table and the consumption rate or predation rate at the same time, you can use toSeq. But you have to include the predation rates or consumption rates. They are the red numbers. If there is no predation for a stage, you have to write a red -1. You can calculate the consumption matrix C from matrices N and C total. Each C total is divided by the respective NXJ to get the CXJ. Once you have prepared your data file, you can run toSeq and click on B read NF button. You will see the message. Are there predation data in this group read live table file? If there is no predation data, you must choose no. If there is predation data, you must choose yes. Then you can open the group read live table data file. You can make sure you choose the correct file in the preview window. You have to enter the time unit of your data. If you collect your data every half day, you have to enter 0.5. When the program asks, do you want to construct individual read live table? Click yes. Convert group read live table to individual read data is important, but not easy. Sometimes it's possible to convert group read live table data to individual read live table data. Then you can use the individual read live table data to run Bootstrap to estimate the variance and standard errors. You can also do the individual read live table to obtain files for the simulation of population growth using timing program. Because the mean fecundity will be assigned to all surviving female adults of the same age, the standard errors estimated by using Bootstrap will be underestimated. If you use 3 replicates for group read live table study, you can convert them first separately. Then you can pull the 3 converted individual live table data to get a more realistic live table. You will see the message, please run toSeq using 083 underscore group read underscore toStage.txt. In order to convert the group read live table data to individual read data, the program will firstly convert the group read live table to a 2-stage group read live table. When you see the message, do you want to group 2 or more consecutive stages to a single stage? You can choose no. When you can see the survival rate SXJ of group read live table, analysis completed. Because the insects are read in group, the program will not give you the mean fecundity. Therefore, the cell of mean fecundity, F, the red arrow, will be left blank. How to calculate the mean fecundity for group read live table? If you know the total number of female adults emerged from this cohort, you can use the relationship between NF, N, R0 and F to calculate the mean fecundity. However, without the fecundity data of individual females, you cannot calculate the standard error of the mean fecundity. When the analysis is completed, a file 083 underscore group read underscore toStage.txt is ready for converting to individual read live table. In order to convert the group read live table data to individual read data, you must analyze the 2-stage group read data using BRead NF button. Message: are there predation data in this group read live table file? If there is no predation data, you must choose no. If there is predation data, you must choose yes. You have to open 2-stage group read live table data file. Enter the time unit of your data. Do you want to construct individual read live table? You must choose yes. The toSeq program will intelligently identify the case of your data. toSeq is intelligent and will give you proper suggestion. In this case, it notices there is overlap between pupal and adult stages, but there is no mortality during the overlap period. It's possible to convert this live table to individual data. In general, you should follow the suggestion. If you see this message, your group read live table is successfully converted to an individually read live table file. You should copy the new data file to a new folder. If the file name of the new file is too long, you can rename it to a shorter one. Analysis and conversion are completed. You can see the survival rate SXJ of two-stage group reared life table. Please check the population parameters. They should be exactly the same as the original four-stage group reared life table. You can analyze the two-stage individual life table following routine procedures A1, A2, and A3. You can also continue to convert it to a three-stage individual life table. In the file, individual reared life table data with two life stages, you can see detailed information for each individual. Because the insects are reared in group, the mean fecundity is assigned to all surviving female adults at the same age. You can analyze the two-stage individual life table following standard procedures A1, A2, and A3. Because the mean fecundity is assigned to each female, the fecundities of females are non-integer. When you see this message, you must accept non-integer fecundity. Once you have completed step A2, you will see the survival rate curves. Because it's an individual life table, the mean fecundity can be calculated. Because it's an individual reared life table, you can use the bootstrap technique to estimate the standard errors. What are the advantages of group rearing? You can save time, space, labor, and money. Females can mate with strong males. The probability that a female doesn't produce eggs due to the weakness of its mate is low. Insects can help each other to digest food. The growth of bacteria will be limited. It might be closer to the natural condition. What are the disadvantages of group rearing? You don't have data for each individual. You don't have precise developmental time, APOP, TPOP, ATC. You cannot use bootstrap technique to estimate the variance and standard errors. You cannot use timing to simulate the population growth. There may be cannibalism or competition. Can you convert two-stage individual life table to a life table with all life stages? Egg, larva, pupa, female, and male adults? The answer is yes. A video will be prepared to show you how to do it. You are welcome to subscribe to this channel. Thank you very much for your attention. We wish you a successful research.